After studying about the sex determination in animals, let us start with another topic, that is sex determination in plants. Now, the mechanism of sex determination in case of plants is almost similar to like that of animals. This mechanism has been studied in large number and various plants. Now, in most of the plants, the sex is controlled by the Y chromosome. This control of sex that occurs due to Y chromosome is almost similar to like that in case of man, and therefore on this basis, if in case of plants Y chromosome is present, then it would be considered or it would develop male flowers. While if the Y chromosome is absent, then would develop into female. This type of sex determination was commonly observed or was first observed in the liverworts. This was first studied by a scientist named Allen, who studied about plant chromosomes that belong to the genus of liverworts. The most common species or the genus of the plant was Sphaerocarps. Thus, Sphaerocarps were the species of the genus plant, that is liverwort. In this case, the male gametophyte had 7 autosomes plus 1 Y chromosome, while the female gametophyte contained 7 autosomes plus 1 X chromosome. Therefore, on this basis it was identified that which plant would possess male gametophyte and which one would be female. On this basis the cross was done in such a manner that parents containing both male and female, when they are crossed with each other, then the autosomes, that is AA, and XY, that represents the male, and in case of female, the autosomes AA plus XX, when undergoes cross and forms the gametes. In case of male, it forms A plus X and A plus Y, while in case of female, only A plus X would be formed. So, when these gametes undergo cross-fertilization, would give F1 generation, that is 1 AA plus XX and second AA plus XY. That is, this will result into female and this male plant, because it contains Y chromosome and it lacks the Y chromosome. From this study it is very obvious that here the male plant contains or is heterogametic in nature while the female is homogametic. In this manner, the homo or heterogametic nature of the plant was studied in various aspects. For example, in angiosperms, let me classify in form of female and male. F represents female and M represents male. So, in case of angiosperms, the female would be homogametic in nature, while males would be heterogametic type. Similarly, in case of other plants like Humulus japonicum, the same would be observed, that is homogametic female. Now that is very obvious because homogametic, that is containing two X chromosome, and heterogametic male, that is containing one X and one Y chromosome. Similarly, it was in another plant named Dioscorea cinnulate, where the same result was observed, that is female is homogametic containing two X chromosome, while the male is heterogametic containing one X and Y chromosome. But there were also exceptions in few plants like the Flagellarian plant, possessed heterogametic female while homogametic male. Thus, in this manner, there are a few exceptions in case of even plants like animals, where the female heterogametic nature would occur in the form of XO, while the homogametic would occur in the form of only Y nature. This was all about the diploid plants. Now, let us study about the sexual differentiation in monoecious plants. The term monoecious we have already studied in the previous section. Monoecious plants means both the reproductive organ, or that is both the male and female flowers, are present on the same plant. One such example of monoecious plant is maize, and therefore maize is considered to be monoecious because it possesses both staminate, that is male part, and testulate, that is female part, both types of flowers on the same plant. Thus, in this manner, the both male and female flowers occurring in different types and at different position would occur on the same plant. Such female flowers, in case of monoecious plant, generally develop along the sides of the stalk, while the male flowers grows at the top of the plant. In this manner, both types of flowers can be distinguished easily. This is in general manner, but in few exceptions, like in case of corn grains, such a development occurs at the tip of the plant. That is, instead of the top, the development occurs at the tip. Now, why does this exception occur? Because of the presence of a mutant gene. Mutant gene is represented by TA. Thus, because of the presence of mutant gene in homozygous condition, that is TA into TA, converts the male flowers into female one. and therefore, due to the presence at the tip of the plant of male flowers, in case of homozygous condition, it gets converted into the female part. But along with this mutant gene, there also occurs another mutant gene which is depicted as BS, and if such a mutant gene reacts with the another mutant gene, that is TA, it forms heterogametic, that is TA and BS. Such a type of heterogametic gene suppresses the development of female flowers. That is, it suppresses the female flower development. And therefore, in this manner, the male flowers does not get converted into the female one and remains as it is. However, in case of other plants, although the mechanism remains the same, but the controlation occurs with the help of another gene. Like in spinach, the sex is controlled by a single gene that is represented by M, and it is located in the X chromosome. Thus, this is all how that the sexual differentiation or the sex determination in case of plants, both monosexual and disexual, occurs.